I'm David Threadgill, Professor of Genetics at University of North Carolina and Professor and Head of Genetics at North Carolina State University. So there's been a number of instances where drugs have passed preclinical toxicity testing, and once they reach the clinic and were exposed to many different patients, then toxicities were discovered. And had we done our preclinical testing much better, we probably could have detected many of these potential toxicities and either identified ways to prevent the toxicities or, more importantly, identified markers that would allow us to properly select the appropriate patients to receive the drugs and to keep it away from the patients that are likely to develop toxicities. The way we approached this, we decided to basically look at how drugs are deployed in the clinic. They're deployed in very heterogeneous patient populations. They're genetically very different individuals. And we rationalized that we needed to model that in our preclinical studies. Currently, most drugs that are being tested by pharmaceutical industries are tested in a single strain of mouse. Now, this is analogous to taking and testing a drug in a single person, which we know that people respond dramatically different to drugs, and so it's not really an appropriate test. With the mouse being the predominant biomedical research tool, we decided to build a population of mice that were very genetically diverse, making it very similar to the diverse patient population that those drugs were likely to find. So the implications for pharmaceutical research are really twofold. One of them being we can do a much better job at detecting drugs that are likely to have toxicities and matching those with the patients that should be getting those drugs. The consequence of that is we should be able to save drugs that may have shown toxic side effects in that one mouse strain that they were tested, but in reality, other patients may get benefit from that drug. And so even though we used acetaminophen as the model drug, it's really more broadly applicable to all drugs and the way drugs are actually developed, both for the process of testing for toxicities as well as testing for their beneficial uses.